Researchers are utilizing gold nanoparticles as a means to combat infections without relying on antibiotics. Greetings, everyone. In today's video, we will be discussing a remarkable scientific advancement. Researchers have successfully devised an innovative approach involving laser-heated gold nanoparticles. This technique not only allows visualization of bacterial biofilms, but also eradicates them, potentially serving as a substitute for antibiotics in combating drug-resistant superbugs. Through experiments conducted on rodent teeth and skin injuries, this method demonstrated swift and efficient outcomes, eliminating the need for broad-spectrum antibiotics. By absorbing light and converting it into heat, these nanoparticles effectively exterminate harmful pathogens, presenting a promising solution for tackling infectious diseases. Without any further delay, let us commence our enlightening discussion. Imagine the possibility of using a laser sword to combat microscopic invaders within your own body, rather than in a distant galaxy. A groundbreaking method has been created by scientists that involves the utilization of laser-heated gold nanoparticles to both detect and eradicate bacterial biofilms. Through experimentation on rodent teeth and skin wounds, this innovative approach shows promise as a potential alternative to antibiotics in the treatment of drug-resistant superbugs. A groundbreaking method has been created by scientists from the University of Pennsylvania and Stanford University, utilizing gold nanoparticles coated in sugar. By subjecting these nanoparticles to near-infrared laser heat, they have successfully demonstrated the ability to detect and eliminate biofilms found in the teeth and skin wounds of rats and mice. Clusters of microscopic organisms that adhere to various surfaces are known as biofilms. These surfaces encompass both inanimate objects, such as stones in a flowing body of water, as well as living components of animals, including the skin and gastrointestinal lining. In a publication in the Journal of Clinical Investigation, the remarkable capabilities of these nanoparticles were showcased, revealing their ability to both diagnose and treat stubborn bacteria and viruses. Mariam Hajfatalian, the study's primary author and a doctorate holder in mechanical engineering, expressed her excitement about the groundbreaking infectious treatment technique that has been developed. She described it as a swift and medication-free approach to imaging and treating skin infections and wounds, with promising potential for clinical application. Using miniature gold particles and light, a novel method has been developed to cleanse teeth and wounds, as stated by the speaker. This groundbreaking approach marks the inaugural utilization of engineered gold nanoparticles in the eradication of oral biofilms through photothermal therapy. In less than a minute, these particles demonstrate remarkable efficiency in eliminating harmful bacteria, all without the need for broad-spectrum antibiotics. The exceptional efficacy of this technique opens up possibilities for the creation of innovative products, such as specialized mouthwash, adhesive strips, and bandages that utilize light to effectively cleanse wounds. The National Institute of Biomedical Imaging and Bioengineering, NIBIB, was among the various branches of the National Institutes of Health, NIH, that provided support for the research. Louisa Russell, a doctorate holder in materials science and engineering and the program director in the Division of Discovery Science and Technology at NIBIB, emphasized in a press release that this platform offers the ability to eliminate biofilms without the need for surgical debridement, a step often required when utilizing antibiotics. This approach has the potential to provide advantages for individuals who are unable to tolerate conventional medications. It is effective even for those who have allergies to antibiotics or suffer from infections that are resistant to common treatment methods. Ms. Russell emphasized the significant advantage of this method being free from antibiotics. The eradication of biofilms was achieved with an almost flawless success rate through the utilization of photothermal therapy. Ms. Hajfathalian emphasized the remarkable expediency of this treatment, particularly in the case of oral infections. In a mere minute, or even less than half that time, the laser effectively eliminates the vast majority of bacteria. In their observations, the researchers expressed their anticipation for additional examinations to determine the efficacy of this approach in cavity prevention and expediting the healing process. To grasp the mechanism behind this light-driven therapy, it is necessary to first comprehend the nature of biofilms. In moist environments, microorganisms come together to form biofilms, which are communities that attach to surfaces and multiply. These biofilms are created when bacteria produce a sticky substance, resulting in the formation of structures such as dental plaque, slimy residue in showers, or coatings on various surfaces, including rocks and even human bodies. According to the Microbiology Society, biofilms can consist of either one or multiple types of microorganisms, such as bacteria and fungi. These formations bolster the survival of microbes by offering them both support and defense against various dangers, including antimicrobial substances and immune reactions. Up to 80% of infections in both humans and animals are attributed to biofilm infections, which frequently impede the healing process of wounds. These infections can lead to a range of problems such as gum disease and urinary tract infections, and have the ability to adhere to medical equipment like pacemakers and contact lenses. As stated by the Microbiology Society, biofilm-associated bacteria have been found to possess a resistance to antibiotics that is approximately 1,000 times greater than that of bacteria existing in a free-floating state, thus attributing them as a significant contributor to treatment failures. Ms. Hajfathalian explains that the intricate network of proteins and carbohydrates found in biofilms can impede the effectiveness of antibiotics. The presence of biofilms poses a significant challenge for current clinical therapies due to the development of antimicrobial resistance. Research suggests that antibiotics alone are often insufficient in treating established biofilm infections, highlighting the urgent need for improved diagnostic and treatment methods. Nanoparticles, which are too small to be seen with the naked eye and range from 1 to 100 nanometers, offer a potential solution. Gold nanoparticles have the ability to absorb light in both visible and near-infrared regions. By utilizing near-infrared lasers, which emit radiation that is harmless to tissues, we can stimulate cell regeneration and enhance blood circulation. These advancements can lead to faster healing processes. Microscopic particles composed of noble metals, which are highly resistant to corrosion and oxidation in humid environments, are known as noble metal nanoparticles. The most prevalent noble metals used for this purpose are gold, silver, and platinum. These nanoparticles have the potential to be utilized in various fields such as drug delivery and thermal ablation, a technique that employs heat to eliminate undesirable bodily tissues. A team of researchers from Malaysia, India, and Saudi Arabia published a review article in Frontiers in Chemistry discussing the use of noble metal nanoparticles as antimicrobial agents. These nanoparticles are highly stable and biocompatible, making them ideal for this purpose. Their small size and large surface-to-volume ratio enable them to effectively penetrate bacterial membranes. According to the authors, metal-based nanoparticles play a crucial role in the 21st century, particularly in nanomedicine and other biological applications. They can be synthesized using different methods and successfully employed in a variety of nanomedical and biological applications. The authors of the study suggest that gold nanoparticles, known for their lack of toxicity, hold great potential in the field of photothermal therapy. These nanoparticles have the ability to convert light energy into heat, effectively eliminating pathogens. Additionally, they have the unique property of emitting ultrasound waves when exposed to light, allowing for non-invasive photoacoustic imaging, a technique used to capture detailed images inside the body. This groundbreaking research may mark a new era in the treatment of infections, potentially comparable to the revolutionary discovery of antibiotics. Ms. Hajfathalian expressed our collective aspiration as scientists to investigate groundbreaking strategies in the fight against infectious diseases. The anticipation of potential breakthroughs fills us with enthusiasm as we eagerly anticipate the progression of our research, which will include animal studies and clinical trials. Alongside her research on nanoparticles for photothermal therapy, she and her team are exploring alternative approaches to decrease the dependence on antibiotics, such as phage therapy. This innovative treatment harnesses the power of bacteriophages, which are viruses that specifically target and eliminate particular bacteria. Ms. Hajfathalian, in her review published in Wire's Nanomedicine and Nanobiotechnology, highlighted the potential of intricate metal nanostructures in personalized therapies. Although awaiting approval from the US Food and Drug Administration, FDA, for clinical trials, Early findings suggest that these nanostructures possess remarkable targeting abilities, biocompatibility, and stability, rendering them highly promising for practical applications in clinical imaging and treatment. In the pursuit of knowledge, forthcoming studies will delve into the efficacy of nanoparticles in combating infections caused by biofilms in various areas of the body, including catheters, heart valves, lungs, and prosthetic joints. Mansoor Amiji, an esteemed professor at Northeastern University in Boston with a doctorate in pharmaceutical sciences, cautions against potential complications associated with the internal application of metallic nanoparticles. Although gold particles have proven to be effective in treating surface infections, there are valid concerns regarding their safety when they enter the body. The question of what happens to the gold once it is inside and whether there are any potential toxic effects associated with its accumulation within the body remains a topic of discussion, as stated by the individual interviewed by the Epoch Times. While photothermal therapy has been used on easily reachable tumors for some time, there is a pressing need for additional research on the safety and wide-ranging efficacy of metal nanoparticles against different organisms, as pointed out by the speaker. According to Mr. Amiji, it is crucial to develop improved antibiotics and explore the potential of non-antibiotic agents for treating infections. He clarified that nanoparticles are not expected to replace antibiotics entirely, but they can expand the possibilities for scientists in their quest to discover effective non-antibiotic treatments for diseases. To support our channel's growth and ensure broader awareness, kindly hit the like, share and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.